So my name is Ben. I'm a specialist biomedical scientist working here in the clinical microbiology laboratory. My favourite subject at school was science, in particular biology. So my job is the interpretation of a variety of different lab tests where I interpret patient samples for the presence of pathogenic microorganisms. The biggest aspect of the job that I enjoy is at the end of the day we can do science and help people, and I think the wide variety of different things that we come across in terms of organisms is one of the most fascinating aspects of the job. So there's always something new to add and there's always something new to learn. I think the biggest challenge is, again, making sure that we're supporting our patients. So at the end of the day, every sample that comes in here is from someone that's probably at their worst point. So we need to take that into consideration in everything we do. So I'm currently studying a Master's in clinical microbiology with the aim of developing my microbiology skill set.
My name is Beth and I'm currently an assistant healthcare scientist. So I did a healthcare science level 3, which gave me a level 3 BTEC in applied science. I actually found out through the government website. I was searching for uni courses because I was looking to go to university. But I found that and I thought it sounded perfect, so I applied and here I am. Prior I had GCSEs and 3 BTECs. The entry requirements were HC in maths, HC in English and HC in science or GCSEs. Science. It's given me a lot of insight in what I want to do for my career, and it's given me the opportunity to actually practice and do what I love before I commit to it. It's a lot of checking samples and processing and culturing samples. Like today, before filming this, I'm doing all the MRSA samples. That's a tough one because I enjoy all of it. I have to say the thing I most enjoy is seeing the after product, like after you've cultured it and seeing all the organisms grow, because they're so colourful, so fascinating, and everything's different, it's never the same. I have to say just making sure that you're doing everything to the best you can. It's really all about making sure that you are doing something that you'd be proud of. Yes, I am. I'm actually looking towards doing a biomedical science degree. Do it. You will not regret it. Apprenticeships are fantastic. I'm glad that they're becoming more and more prevalent, especially in this field. When I was, younger, these were few and far between, and those that weren't as favoured by universities, but now the experience, the qualifications, it's all becoming much more accepted. And actually it's a great way to learn, get money at the same time, and it's a fantastic opportunity, so if you get it, try it.
Sample processing starts with labelling of samples, double checking the name and details of the sample, and then deciding what tests need to be done based on which ones have been requested and whether or not the specimen is appropriate for that test. The sample will then be taken to the most appropriate area of the laboratory for processing. So in this case, Ren's deactivating COVID-19 samples for COVID PCR testing in this cabinet. For swabs and samples taken through bacteriology, we place the samples onto culture media, which enables us to grow the organisms up and identify if any pathogens are present. So another one of our apprentices here is inoculating culture media for the isolation of single colonies that can then be identified tomorrow morning when we grow up the culture plate in a 37-degree incubator overnight. So here Matthew is culturing some tissue samples. With these we go one extra step and we also inoculate a liquid broth to then isolate any more fastidious organisms that might not grow on the agar plates at 24 hours. So we place our plates into the incubator and then we leave them for 16 to 18 hours and then we'll see what grows on the next day. So here's just a small short example of some culture plates that have incubated overnight, and as you can see there's a wide variety of different organisms that we isolate. So these are from wound swabs, so these could be anything from ear, eye, mouth, nose, you name it, it can go onto this bench and we'll try and grow something from it. When we do grow something, the BMS or Biomedical Scientist then needs to make an interpretation as to whether or not what's grown is significant. So what I'm doing here is I'm looking through the plates and judging as to whether or not anything warrants further work or follow-up. So to identify the organisms we've grown on the plates, we use a giant laser, which is called a multitof. However, there's lots of other different ways we can also use to identify these organisms, such as going on what they look like on the plate or what sugars they might use to grow. One other really important method we use on a daily basis in the lab is microscopy, and we look at gram-stained bacteria that show up as gram-positive blue or gram-negative red. So here you can see some gram-positive cocci, which might be Staph aureus, for example, the organism that's associated with MRSA. So after we've grown an organism and decided that it's significant, we then need to figure out what the best antibiotic is to use to treat this. So one of the methods we do here is we inoculate a plate that's got a whole range of antibiotics at different concentrations, and then we use that to determine which one we can use to effectively treat a patient. So in the past, a lot of these tests were done manually, and we still do a lot of them manually ourselves, but nowadays the technology is there where we can automate a lot of these tasks. So here's a recent example of us automating a COVID-19 PCR master mix setup. Another recent advancement in automation within our laboratory from a couple of years ago is the automatic spreading of MRSA plates. So this machine here will place the swabs and spread them for colonies onto the plates completely automatically. All we need to do is load it up and walk away.